Now that we've talked about the individual nucleotides, we'll talk about stringing them together. So the nucleoside triphosphates are the building blocks of putting together a string of nucleotides. We already saw the individual bases that would attach to the nucleoside, and then the triphosphate is going to be the energetic form of it that will be able to be the monomer that you add to the chain. The chains aren't just made from scratch. You have to have another chain as your template and build a new chain by pairing with the previous chain. So the cell will have to maintain pools of each nucleotide triphosphate or deoxynucleotide triphosphate. So there's an enzyme like this ribonucleotide reductase, which can form the deoxy version from a ribo version. And there's another process to convert uracil into thymine. So the cell has to maintain the proper levels of each nucleotide, either the diphosphate, triphosphate versions, or the deoxy or the ribo versions. And one way to control the rates of, say, DNA duplication is to control how many nucleotides are present in the cell, because the DNA duplication process or replication process is kind of just waiting it for each nucleotide to kind of randomly bump into where it's trying to build the chain. So the fewer free nucleotides, the slower it'll go, and the more nucleotides, the faster the process will go. So the primary structure of any nucleic acid is going to be the sequence of bases on the chain there. So there's the notation of just listing the bases, and then if more complicated situations, you might have a code meaning that there's a set of different bases that might go in the location. So once you have a string of nucleotides, they can start forming double-stranded or various helices, various structures, and analogous to the protein structures of primary, secondary, tertiary, and quaternary. So you can see here this chain of nucleotides. You have the phosphate sugar backbone of either DNA or RNA, and then the bases attached to them. And then these bases, this is a single-stranded version. The double-stranded version would have the anti-parallel sequence in the other direction, forming hydrogen bonds between the strands. So in the sequence, if you have a pattern that kind of shows up in multiple places, you can start describing it as a sequence motif. So there's kind of various ways to describe what is showing up there. There's the sequence logo where you kind of list out at a certain position what is most likely to be found at that position. So you can see that in certain places there's kind of a somewhat more even distribution of values, and then at certain places there's almost always a single type of nucleotide. And the y-axis is the bits of information or information content. So since there's 4 bases to choose from, since you already know it's either RNA or DNA, so there's no conflict between uracil and thymine, you know that each position has 4 options, and 4 options means 2 bits. So if there is a position where you're just as equally likely to find any of the 4 possible nucleotides, there'd be kind of no information at that position. And if there's a position where you can almost always see the same nucleotide, then you need kind of 2 bits of information to tell somebody that that's the one out of 4 specific base that you'd see in that position. And I guess that's pretty much everything I was going to say about kind of information content for a sequence of nucleotides. So all the domains of life use DNA and RNA in various ways. It's a universal kind of information content storage or information transfer going between DNA and RNA and performing functions. So there's kind of alternative classifications of life. So you kind of have here one possible classification is splitting it between cellular and non-cellular life. So what I was kind of mentioning was cellular life, the various domains of, mostly archaea, bacteria, and eukaryotes. But there's also the viruses that can be single-stranded DNA or RNA, or double-stranded DNA or RNA. And they're not always, I think they're mostly never classified as life, but you can think of them as non-cellular life. And this kind of is the total group of entities on Earth that use DNA and or RNA. Just to mention real quick, once you have a strand of DNA, especially a double-stranded DNA, you can start getting into various twists of supercoiling. So there's a lot of like tension in the DNA and ways to kind of twist the double-stranded DNA to add or remove tension, and that has to do with freeing up some tension during the replication process, and I guess kind of organizing the DNA in the chromosomes and various things. And then to mention about RNA, RNA is more of a temporary storage of information. So this here is a way that it's kind of eukaryotes can degrade RNA. So you have DNA as the storage of information that's a permanent storage. RNA is a copy of the information with the explicit intent of having it be a temporary copy that has some degradation rate. So that's kind of like, I guess, the main difference between DNA and RNA is DNA is a more permanent storage of information, whereas RNA is more of a time-limited copy of information. And DNA being permanent storage of information still has different means of expressing the information. So, every differentiated cell in the body has the same DNA, same information, with additional information deciding on when and where in the DNA to express the various genes.